Okay, I'll be opening up the room. Just give me 30 seconds and then I'll welcome everybody. Good afternoon, everyone. Today's speaker is Dr. Wei Yang.
Let me adjust this thing a little bit. Okay. Oh, good afternoon. And so today I'm going to talk about congenital birth defects. And so this condition is mostly caused by genetic mutations or genetic changes. So of course, epigenetics can also be involved. So that's not the focus of this lecture. So my name is Wei Yan, and so I'm an investigator at the Lundquist Institute of Biomedical Research, Biomedical Innovation at Harvard-UCLA Medical Center. So I'm also a professor of medicine in the Giffen School of Medicine at UCLA. So before I get started, I try to get the the baseline knowledge of you guys. And so if you don't mind, we can take this so-called pre-lecture test. So you look at your screen, choose the answer you think that's the right one, and let's see how it goes. So take 10 seconds. So let's see what the results look like. All right, so you guys good and good on this first one. Okay, so let's do the next one, please. So pick one you think that's right. Okay, so can we see the results? All right, so 79%. The first one, 87%. So let's get started. Let me see. Okay. So when we talk about birth defects, as I said, most of the birth defects results from changes in the gene structure or the chromosome numbers, all the changes at the genetic level. So in order to understand the genetic causes, it's important to know the definition of a gene. So actually, the concept has been evolving, but the basic, the original definition is like this. So you think about it, our body. And so if you think, it really depends where you set the bar is. So if you think there's a basic functional unit, so you can say a cell, right? So this is most of us think about it. So if you look at the cell, and then you have the, like a cell like this. So this is cytoplasma, where you have all these organelles. And then in the middle of the cell, usually, and you can see a structure called the nucleus. So if you really get into, like a zoom into the nucleus, and actually the nucleus consists really is occupied by the structure called chromosomes. And then these are actually the a large molecules called DNA. And so deoxyribonucleic acids. So they form this long double helix strands, and then they wrap around the histones and form the chromatin. And then through condensation, so they form a chromosome. So this is basically what's inside the nucleus. And if you further open up this chromosome, as I said, you see this long strand of molecules called the DNA, and then they wrap around the histones to form the basic functional unit called a chromatin. And then if you really further open up the DNA, you're going to find the DNA actually consists of 4 bases. So namely, adenine, guanine, and cytosine, and thymine. So these 4 bases actually they can pair up. For example, like A preferentially and pair with T, and C preferentially pair with G. So that's why you see this kind of like pair, like the bases paired and form this double helix structure. So the definition of the gene is that the one gene contains many of these nucleic acid pairs, and then those bases actually the order, the organization of these 4 nucleotide bases actually represent the genetic code. So in other words, the traditional definition is that the stretch of DNA that can, carry the genetic code that can guide the synthesis of protein. But the new definition is really, because if you look into the human genome, the vast majority of the DNA, they are occupied by sequences that do not encode proteins, and the so-called the non-coding regions. And actually, the coding regions only account for a small percentage. So the previous definition is that the genes refers to coding. I mean, like the coding sequences on DNA. So in other words, if you have a stretch of DNA, but the sequences do not encode protein, they're not counted as genes. But the new concept actually is changing, because these days we know that they may not encode proteins, but they do have functions, most of them, okay? So if they have function, they should be considered as gene as well. So that's, I don't want to go into too much details, but the take-home message is that, as I summarize in these slides, so the genes referring to those DNA sequences that can guide the synthesis of protein is sort of like a command center within the cells. And then based on the genetic code, messenger RNAs are synthesized, and messenger RNAs are further translated into proteins, and the proteins actually form the network, like a functional network, to execute different physiological roles. And so basically, the DNA is regarded as the instruction book and to regulate the cellular function. And so in human cells, typically each cell contains the 23 pairs of chromosomes. Remember why we talk about pairs? Because one set comes from mom and the other set comes from dad. So basically it's a paternal, maternal copy of the chromosome. That's why you have 23 pairs. But there's a difference between men and women, because in men, so both men and women, we have 22 pairs of chromosomes called autosomes, and so, but we do have a pair of sex chromosomes. So in women, there are 2 X chromosomes, and then in men, so you have 1 X chromosome, 1 Y chromosome. So now if we go back to the question we just had, and I just think that we don't have to do this thing, and because 87% of you actually did it right. And so all these descriptions of number 1, 2, 3 are all correct, and the fourth is wrong, right? So men, women, and the two sexes, they have different sex chromosomes. So that's why not exactly the same 23 pairs, right? So that's why. So hopefully that 13% of you now know the correct answer. So let's move on to birth defects. So what's the definition of birth defects? Like, the name actually already told you, right? So the defects you can see at birth. Actually, it's not really true. So basically, birth defects refers to structural, behavioral, functional, metabolic disorders present at birth or soon after birth. It's also called congenital malformation or congenital anomaly. And so there's a specific subject, like in biomedical research, called teratology. Basically, this is the field that specifically study the birth defects. So how about incidence? The incidence of birth defects is about like 2 to 3% of all the live-born infants. And this is like the defects that can be recognized at birth. An additional 2 to 3%, you cannot recognize at birth. It's not that easy because those defects are sort of like subtle, but you can find out, actually realize that within 5 years after birth. And so in terms of the mortality rate, the birth defects account for 21% of infant deaths. And then there's no ethnic differences. And so in terms of causes, and basically it can break up like this. So 50% unknown, so we don't know yet, but 15%, and we already know that. So that's like those birth defects results from known genetic factors. So for example, chromosome abnormalities, which we're going to talk about mainly the next couple of slides, and then gene mutations. And then there are 10%, and they seems to be caused by environmental factors, like environmental exposure, like exposure to the toxins, or certain diseases, or like you take some drugs during pregnancy, things like that. And then 20 to 25%, they have these combined causes, like genetics plus environmental factors. And that's one thing is kind of interesting. Twinning actually is a major contributor to birth defects. So the more fetuses you have simultaneously, they're competing for the limited support, and so from the microenvironment of the uterus, and then so that really increased the chance of birth defects. So remember, here's the thing: so 50% of newborns, they have this minor abnormalities. This is like some of them like a really minor, but there's always some correlation. So if you have so for example, if you find one minor anomaly in the newborn baby, so you have 3% chance of a major malformation you're going to find out later on. And then so the same thing, if you have 3 minor anomalies and identified at birth, you have 20% chance of several major malformations, maybe within the internal organs or some major organs. So types of birth defects. So there's the first one is the malformation. So malformation occurs during organogenesis. So basically that's during the 3rd and the 8th weeks of fertilization. Development. It refers to a complete absence of a structure or alterations in the normal configuration. So basically, it's not formed correctly. And a disruption. So disruption means the morphological alterations of already formed structure due to a destructive process, like if you have a vascular accidents and you're gonna that further development is prohibited. So you're gonna have this like, so for example, the bowel atresia, and you're gonna have a defective development in certain organs. And then deformation. So usually this refers to some sort of like a physical process, like mechanical forces that mold a part of the fetus over a prolonged period. And this is like you have everything, but because of the obstruction or like a certain mechanic forces that change the morphology of that organ, if you will. So that's why this type of a birth defects sometimes can be reversible postnatally. So if once the mechanical force or these obstructions are like removed or disappear, so the morphology can resume to close to normal. And another type of birth defect called a syndrome. So this is something like really hidden. So you can identify a group of anomalies occurring together due to a common cause. But the thing is that usually, in clinics, you identify a number of symptoms, a number of anomalies, and then later on, if this all these problems, like the defects identified always together, so that it probably results from the same cause. So that's why people call them syndrome. So as I said, remember I told you that 15% with the known genetic causes, and now you have about 10% caused by environmental. So the environmental factors can cause genetic defects, and but the thing is that you have these major defects during development is highly unlikely is caused by environmental factors. I mean, unless this environmental factor acts on the mom. And so basically, as I said, my lecture, we're going to focus mainly on the genetic causes. Okay, so basically, we're going to introduce you some, major genetic changes that can lead to birth defects. So the first one is the so-called numerical chromosomal abnormalities. So this refers to you have abnormal number of chromosomes, and so it's mostly caused by so-called non-destruction. And then the non-distinction meaning that, when chromosomes segregate and they don't segregate correctly. So I'm going to show you an example in the next slides. So this can happen during meiosis. Remember one important thing. So meiosis is unique to germ cells only, okay? So your somatic cells, they undergo mitosis, and meiosis only occurs during germ cell development. So during germ cell development, what are germ cells? So in males, it's spermogenic cells, and then the final gamete is called a sperm. In the females, you have the oogonia, like an oocyte, and that's the female gametes. So that's the germ cells. Okay, so during the spermagenesis and oogenesis, and those germ cells undergo meiotic cell division. So if non-disjunction occurs during meiosis, and you're going to end up with the abnormal number of chromosomes. But that because your germ cell chromosome composition is changed, meaning that you're missing one chromosome, you'll have an extra copy of chromosomes. And then after fertilization, if that gamete, like either sperm or oocyte, it participates in fertilization, and the offspring, every single cell in the offspring will contain exactly the same abnormal number of chromosomes. Okay. However, the non-disjunction can only occur, can also happens to mitotic in somatic cell during mitosis. So if that's the case, you're going to have this so-called mosaicism. So basically, it's like a certain number of cells, so for example, in one organ, and certain percentage of cells will have the abnormal number of chromosomes, whereas the remaining cells within that organ, they have normal chromosome composition. Does it make sense? So, if it happened to during meiosis, so the abnormal number of chromosomes will remain in the gametes, either sperm or oocyte. So if sperm fertilize a normal egg, or if a normal sperm fertilize a egg containing abnormal number of chromosomes, and then the resulting zygotes, and then later on they develop into embryos and then develop into individual. So these individuals, every single cell will have abnormal number of chromosomes. Versus if it happened during mitosis, because it's within the somatic tissue, it doesn't, it's only cause a certain number of cells containing the abnormal number of chromosomes. So the second main cause is the structural chromosomal abnormalities. So we know that, as I said, in each cells, in human cells, you have 22 pairs of autosomes and 1 pair of sex chromosomes. And then they have different sizes. You have the long arm, you have the short arm. So they have this unique morphology. So if you practice enough, and then once you do the chromosome spread under the microscope, you can accurately identify each of the 23 pairs of chromosomes. So you can, based on just like the size, the shape, you can tell this is chromosome 12, chromosome 21, X chromosome, Y chromosome, and so on. But if during cell division, especially during meiosis, and so because of hormonal recombination, we're going to talk about that. I'm going to show that thing. But the thing is that it's not like all the chromosomes they duplicate and then they separate during cell division independently. No, it's like the homologous chromosomes, like the copy from the mother, the copy from the dad, like the paternal and maternal copies, and then they can get together and then do the exchange of genetic materials. So specifically during meiosis is called a crossover. Actually, the nature of the crossover is a homologous recombination, meaning that the fragments of the chromosome and that they have the similar sequences, they can switch. So between the paternal and the maternal copies. And then during this type of events, if anything happen, like some disruptions occurs, and then you're going to have like a certain, like a small fragments of chromosome, and then sort of like translocated to another chromosome. So the end result is that, actually this translocation, inversion, that they change the orientation, duplication, meaning that they duplicated and they're supposed to segregate, but somehow like they retain there. So you have double copies, 2 copies of the same sequences. And the deletion, of course, you have a big chunk of DNA missing. So these things all can happen if it happened during meiosis, as I said. So although you have normal number of chromosomes, but structurally they're different. So for example, if a chromosome, let's say 21, chromosome 13 or 14, looks like under the scope, you can tell if there's an extra piece of a chromosome from somewhere else. You're going to find out, certain chromosomes becomes longer and certain chromosomes become shorter, and then this is a translocation, because normally it shouldn't be this long under the scope. Okay, so remember, these changes occurs at the chromosomal level. It's huge. So basically, you have the big chunk or the entire chromosome missing, or you have an extra copy of a chromosome, or you have big chunk of chromosomes that go to the wrong place, or go missing, or go duplicated, or sometimes change orientation. So these are really dramatic changes. And then if those changes, and remember, we are missing 1 chromosome, for sure, you're going to lose the function of the chromosome, right? So usually it's very, very severe. But in the second scenario, I just mentioned the structural abnormalities, and you're going to have this, structural changes, but the numbers are correct. And so basically, if this translocation, inversion, duplication, if they don't generate any functional disruption, so they have the individual will not have problem. Otherwise, that's called a balanced translocation. But if they do induce disease phenotype, they call it unbalanced. So commonly occurs in this, couple of chromosomes, 13, 14, 15, 20, 21. The reason is that during a homosexual recombination, so these chromosomes actually tend to cluster together. So when they get to, they are too close. So usually this type of abnormalities, all these disruptions occur among them. So that's why you commonly see this type of problem. And then, as I said, this is like a chromosomal level changes. And then the disruption can also, disruption of gene function occurs also at the single individual gene level. So for example, you only have a change of one nucleotide, one base of nucleotide, and then that could cause a huge disruption of the gene expression. So that's like really occurs at the individual gene level. I just mentioned that meiosis is unique to germ cells. So for example, in the primary oocyte or spermatocyte, and after duplication, so they need to divide. So the hallmark of meiosis is that the paternal maternal copies, and after duplication, they align like this. So they form this so-called tetrad. And then remember here, so as I said, if they share some similar sequences and then they tend to exchange their genetic materials, normally it should happen like this. So you have a complete segregation of these two copies, and then after some changes of certain fragments on the chromosome, and then after these cells undergo the second meiotic cell division, so you have this each one has a single copy. So that's why the cell called, they are, in terms of the chromosome composition, they're haploid, meaning that they only have half the total number of chromosomes in the gametes in either sperm or oocyte. So this is the normal situation. However, if you have non-disjunction, so they may undergo chromosome like a crossover recombination, but they don't separate during the first meiotic cell division, like a meiotic cell division. So you end up an extra copy in the gamete, and then the half the gamete you're going to have like an extra copy, half the gamete they're missing one copy. So if these gametes got fertilized, you're going to have the individuals with extra copy of chromosome. These individuals, if this gamete undergo fertilization, and then the individuals will lack one chromosome. So this non-disjunction can occur during the first meiotic cell division. It can also occur during the second meiotic cell division. So for example, the first division, everything separate normally, but during the second meiotic cell division, they don't separate properly. Again, the same thing. So half the gametes, you're going to have extra copy, half the gametes, they're missing one copy. So if fertilization occurs, that leads to the abnormal chromosome numbers. So as I mentioned, that under the scope, if you do this like a chromosome spread, and you can clearly see these 22 pairs of autosomes and one pair of the sex chromosomes. Based on their morphology, you can tell the chromosome numerical and structural abnormality. So for example, in this case, so you have extra copy of 21. So this is definitely a Down syndrome. And so you normally should have 2 copies of 21. Now you have 3. And then if you look at this chromosome spread, and if you look at here, so the chromosome 14, normally they should be equally, they should have like both copies should have an equal size. And clearly you see there's an extra like an extra segments of chromosome. And actually this is a translocation. And then if you examine the 21, it becomes shorter. So basically it's a translocation between 21 and 14. So these individuals also have basically extra copy of 21. So 21 is extra copy. So this is also a Down syndrome, but caused by translocation instead of this type of thing, like the just pure increase of the number. So I cannot believe I already we're approaching 30 minutes. But anyway, so I just show you that's the main. If you understand that previous 2 pictures I just show you, and then you're done. So basically, all these big, like a common birth defects, like a genetic diseases, mostly involved in the numerical or structural changes of chromosomes. And so this trisomy 21, as I mentioned, the Down syndrome, there's so many features. I don't want to go through everything. And so the incidence is about 1 out of 2,000 newborns. And So I mark the age. This is the mom's age. Okay. So if a mom gave birth to a baby and there's a mom's age younger than 21, the chance is 1 out of 2,000. So if mom's age reached to 35, so 1 out of 300. So if mom's age over 40, and then it's 1 out of 100. As you can see that with aging, and so the incidence of Down syndrome increases dramatically. So that's why for all the women who get pregnant after age 40, they are routinely screened for Down syndrome, because 1 out of 100 is literally 1%. If you don't screen, you're going to have 1% of newborn babies, they have Down syndrome. So that's why, the screening is very, very important. So so this is the numerical abnormalities. Remember, it happens to 13, 14, 15, and 21, 22 chromosome. So now you have this trisomy. There's only 2 types of trisomy. This trisomy 18, and this is a much more severe, numerical chromosome birth defects and caused by the abnormal number of chromosomes. And so if you are interested in looking into them, so but they can be born, but very rarely do they survive. So they usually die. So for example, in this case, within 2 months, and then this will die. 90% of infants will die within a month. So, seriously, I mean, like literally, they're not. This is non-survival. But Down syndrome, though, they can survive and so live for a long time. And so, of course, these numerical changes, like in the chromosome numbers, can occur in sex chromosome. So if it happen in sex chromosome, in this case, you have extra X. So this type of individuals, they displayed a number of problems, and so we call it Klinefelter syndrome, and named after the physicians who first discovered these birth defects. And so there's so many features. As a matter of fact, and the Dr. Wan, Dr. Swerloff, they have been working on this disease for their entire career. So they know a lot more than I do. But the causes, and really the underlying cause is the abnormal chromosome numbers, and mainly occurred during meiosis. All right, so this is like this Turner syndrome. Usually, basically, as I said, normally for females, you should have XX, so 46 chromosomes. So if you're missing 1 X chromosomes because the non-destruction, and then you're going to have this so-called Turner syndrome. Okay, so we're going to skip all these things because of the time limit. So the structure abnormalities, and as I said, you can have translocation, inversion, duplication, deletion. So if you're interested, you can really search the YouTube, and then there's many movies and really short video clips showing the birth defect caused by chromosomal deletions. And this is a large chromosome 5 short arm deletion that cause the this protege. Basically means that the baby is born and then they cry. This sounds exactly like a cat. So so you can check it out. And then the microdeletions, if it occurs to on the chromosome 15, the long arm, and so depending on which copy, if it's paternal copy versus maternal copy, and then they cause 2 types of syndrome. So that's the interesting thing, because it's the same region on the same chromosome, depending on the origin of that chromosome, and they can this type of deletion can lead to different syndromes. As I mentioned, the cause can be environmental, and so viruses, radiation, drug usage, and then so on. So, gene mutation, as I mentioned, like the structural numerical, that's really occurred at chromosomal level. And then, the changes in genes can occur at the individual gene level. So if you have a, single gene mutation, and actually a single gene mutation accounts for 8% of all human malformations. And so remember, you have two copies. Each gene you have like a, you have two allele, and then one's like the paternal allele. Remember, you have a paired chromosome, right? So you have one copy from mom, another copy from dad. So any genes you have two copies. If one copy you have mutation that leads to disruptive function, and then show the phenotype is called a dominant mutation. And if you need two copies that lose their function simultaneously, so that type of mutation called recessive mutation. So again, I remember, I think that 79% got wrong this got right with this question. So obviously now you know that the trisomy 21 causes Down syndrome. Okay, so Klinefelter syndrome is caused by an extra X chromosome, or sometimes even two copies, extra 2 or 3 copies of X chromosomes. And Turner syndrome is caused by missing of 1 chromosomes. And Crotochet syndrome caused by the microdeletion of the short arm of chromosome 5. So I just want to stop here. I don't want to really talk about. So this if you know the causes, so that means you can develop prevention strategies, right? And so you can also develop a diagnostic testing. So if you're interested, you can check on the YouTube, and so some obvious defects and structural defects, and you can fix them when the fetus is still like developing, right? So this is called a fetal therapy. And then if we know the single like a gene mutation, they have a single nucleotide mutation, and then based on the current technology, and that allow us even to fix that genetic mutation. So that's really the some possibilities in the future in terms of the prevention of birth defects. So I just want to stop here. Still go over about 7 minutes. So now we can get started with your question or general discussion. I don't know if you noticed that Kylie is our intern, and she's here as well. I think that Kylie will moderate the discussion. So,
hello, everyone. Hi,
yeah.
Thank you, Dr. Yan. Wonderful presentation. So let's go with Grace. Can I ask a question?
Yeah. Thank you so much, Dr. Yan. I really enjoyed your lecture. I was wondering, so why, I know that some plants can be aneuploid and polyploid, like strawberries have 8 sets of chromosomes. So why are plants so resistant to the harmful effects of like polyploidy, whereas like humans can't really resist that?
Oh, that's a difficult question, and I don't know much about plant genetics, and but the only thing I know is that they're as fascinating as the mammals and all these eukaryotes. But anyway, so I don't know. I think that this is like doing evolution, and if you believe in evolution, so this is sort of like a fail-safe mechanism, and so you have to sort of like really control the number of chromosomes in order to fulfill the like so that they can function correctly. So I think that in plants, remember again, it's going back to evolution. So they have to adapt, just like animals and humans and then plants. They will adapt as well. So that involves many mechanisms, and these things actually these days many of us believe that you have the try to adapt to the environmental changes or challenges, and so you're going to have this epigenetic mechanism. So if certain epigenetic changes frequently, so that might induce certain genetic mutation. Like really, it's really the genetic mutations that drive evolution. So you have some mutations, and that may generate some favorable traits, and that traits actually support the survival of the individual. And so based on the Darwin's evolution theory, and those individuals will have a better chance to survive. So that kind of genotype will be will get propagated and then become dominant. So I don't know, maybe plants that they are facing more harsh environment. So, you know, I don't know if that answers your question or not.
Yeah, it does. Thank you.
Yeah.
Thank you, Grace. Let's go with Kamal, please.
Hi, so why is it that the instances of Down syndrome would increase with the mother's age exactly?
Yeah, that's a good question. So aging has a huge effects on follicular genesis. Remember, so so for that's in women, and when a baby girl is born, so in her ovaries, the number of female germ cells is fixed. So in other words, you only have each girl as soon as she was born, and she has a certain number of follicles containing the oocytes. And so, and then during this month, upon puberty, during monthly cycle, so you have certain cell, usually it's about a dozen follicles are recruited into the proliferation pool, so undergo these monthly changes. Like eventually, you only have one oocyte got sort of like ovulated, got released. And so remember those female germ cell oocyte, they are sort of like cell cycle wise arrested, and then when they are recruited and undergo follicular genesis, they resume the cell cycle. So basically, this hormonal recombination not completed yet. So with aging, as I said, maybe the microenvironment changes, or maybe some like the regulatory networks and the precision of it, that the regulatory network is not as good as before, and sometimes the meiotic cell division, like the crossover hormonal combination, can go wrong, and so that lead to the aneuploidy, that lead to the abnormal number of chromosomes. So aging clearly is a factor. So in terms of the what's the underlying mechanism, it remains not very clear. But one thing is for sure, that aging plays a huge role in the quality of the eggs, and especially regards to with regards to the aneuploidy. So a magic number based on the current literature, that's 37. So 37 years old. And so as soon as you hit 37, I mean, all the women hit 37. So the quality of the egg, so meaning that the chromosome numbers, chromosome structures, and the expression profiles, and the oocyte, and if you do wide panels of examination of either the genome or epigenome, all indicators suggest that after 37, so the egg quality go downhill drastically. So that's why if you can figure out a way that can prolong this aging process, and that's going to be huge. And so, and for some women, you have the so-called premature ovarian failure. So basically, that as I said, like 37 is the number where you start to see the egg quality going downhill. But some women actually, this happened prematurely, meaning that maybe they're in their 30s. Sometimes you can see that patient in their 20s, and they already exhausted their follicular reserve, and the quality is actually going downhill rapidly. So any measures, I mean, of course, you have to know the mechanism. If you know the mechanism, you can prevent that from happening. That's going to be huge. So maybe in the future, some of you guys can solve that problem.
Awesome. Thank you, Vivian. So we're going to do, I tried to remember the order. So we have Devon, and then I think Miss Jones, and then Bianca, and then Risha. I'll put the order of asking questions in the chat as well. But yeah, Devon, take it away.
Thank you so much for your presentation. It was really interesting. My question was, I heard you mention that scientists can fix structures in developing fetuses. And I was just wondering, how does that work, and what can they fix?
Yeah, so let's say if you have remember I told you that different type of birth defects. So for example, you have the deformation, and then so like malformation. So I mentioned that malformation, disruption, and deformation. And so if you identify, and these days actually you can use you put a scope inside the mom's womb, and then so you can really look at the fetus. If you identify any structural defects, you can fix it even during development. And so genetically, if you fix it, so that's like still people are debating. Actually, in reality, some companies are trying to, if you know that a condition, and this is the things that you have to do some testing beforehand, right? So you can get some the fetal cells from the amniotic fluid, and then you can do this genetic testing and try to see if there's any genetic mutations in the fetus, and then using the so-called like this gene editing tools, either talents or CRISPR-Cas, and that you can design a strategy that can replace the mutant genes. So that's still challenging. I'm talking about single gene, like single nucleotide mutations, and but for example, in the case like you have missing the entire chromosome, or you have a big chunk of chromosome missing or inverted. It really depends. So depends on, so for example, if you know this is a balanced translocation, you don't care because functionally they are just like normal, and but they may have some like a minor effects and minor problems. And for this unbalanced, meaning that they do have that thing. And at this point, so to manipulate a specific gene to change a couple of nucleotides is such an easy thing to do. And then they can basically design this CRISPR-Cas, make all this construct, all this guidance RNA, deliver it to the baby, and then hopefully to the target organ. Hopefully they can change the gene to rescue the phenotype. So that's the idea. But nobody has reported a successful case for fetal therapy. So I mean this is like either surgically to repair certain physical structures of the fetus, or you can fix their genes using this latest genome editing technology. But there are a whole lot of limitations. And then the most effective way is prevention. So if you know the mechanism, you know the detection, the early detection, that this is non-survivable, or if pregnancy continues, you're going to end up with a baby with Down syndrome or some very severe birth defect, and that pregnancy may be stopped at a very, very, very early stage, so that you don't have to give birth to a baby with severe birth defects.
That's really interesting. Thank you so much.
Thank you.
So next is Ms. Jones. Ms. Jones, you want to ask a question to Dr. Yan?
So you have to unmute yourself. Seeing that you're muted.
Hi, Dr. Yan. Thank you so much. What a great presentation.
Thank you.
I did have a question. I wanted to find out, a lot of women are having babies older now, just getting their careers and having babies older. What was there a significant change in, because, I know in the, way back in the day, I remember a lot of older women having children. My grandmother was 40 when she had my father back then, but I was just wondering, like, I know African-Americans have a low infant mortality rate and low birth weight, and they believe it's due to the Aztec load and stress and this and any other, but was it more prevalent for Down syndrome babies back then, like say 50, 60 years ago, or is it something that has evolved over this time and we started noticing it more in older women with birth defects?
Yeah, so I think that you raise a very important question, and I'll tell you this. And so my advice to all women is to have children as early as possible. I'm not saying that you're like a normal age, just follow the biology. these days, actually, people try to say, because of career, I want to, set up my career and then I will have children. But we haven't figured out a way to turn the biological clock backwards, sort of like accommodate your career ambition or anything else. So you're going to see a whole lot of advertisement. They're saying that, you're a career woman, come here, we're going to freeze down your egg. And then whenever you are ready, and then later on, we can thaw your eggs, and then we can do in vitro fertilization, and then you can carry your baby, and or you can do this surrogate. So you can find a sort of like another woman to transfer the embryos to her uterus, and then I'll tell you these things. Actually, I strongly disagree. And actually, it's so bad. And remember, you can freeze down your eggs when you are young, but the thing is that with aging, everything else, because it takes not only eggs and then fertilization, and then you have to prepare your uterus and the endometrium, so that you have the perfect environment for the embryos to develop.
Exactly.
And but you have a younger egg, and the fertilization transfer the fertilized egg, these early embryos to a older uterus is not going to work. So that's the point. So your second thing, actually, you mentioned that is because the incidence of all the birth defect increase with aging, or is. I really think that's one contributing factor. That's like when people have their first child, so they tend to be older these days because of career or financial, whatever thing. And then the second thing is that because of the detection. So previously there's no diagnosis, and so whatever baby you give birth to, you have to have that. These days they have this so-called a PGD, if you go through this assisted reproductive technology. This is actually the third generation of assisted reproductive technology. Refers to the PGD, meaning that pre-implantation genetic diagnosis. Basically, right after fertilization, before they transfer the fertilized egg, the early embryos into a uterus, they do the genetic diagnosis. They only transfer the embryos that are free of major genetic defects. But if you go through this natural conception, these days there are many technologies, and there are new technologies, a very sensitive, very specific one, and then they can draw a small amount of fluid from the uterine cavity, or they can do some samplings, and from the chorionic villi, that's represent the the tissue of the fetuses. They can do much more accurate genetic testing. So meaning that you can sequence the entire genome of the fetus to see if there's anything wrong, and if there's major birth defects, they're going to tell you. So what I'm saying is that aging, that's one contributing factor why you see but the advanced genetic testing can prevent this type of pregnancy, so that you don't see huge increase of major birth defects. Although people are postponing their age of pregnancies. I don't know if that makes sense.
No, that made a lot of sense. And I know we have a lot of other questions to go on to that, but I can probably ask, you can answer it to later at another time. But if they've had other babies, like, a lot of times in the old days, they had like multiple babies, and that wasn't their first baby by the time they got to be 50, right?
Yeah,
but they were still having babies late in life. And that might have changed something, or would that have changed something? I know you have another question to go to, but excellent presentation. I have a lot to talk to you about. But
definitely.
Awesome.
So I'm a reproductive biologist. I know a lot. I did nothing for the past 20, 30 years, but study of reproduction. And so I'm also the editor-in-chief. I mean, I just finished my term of the journal called Biology Reproduction. So if you have any questions concerning fertility and reproductive health, and reproduction right, and let's talk. You're welcome to talk to me.
I would love to, because I used to work with Michael Liu and with his, with the CCHN project and all these other with Black Infidels. So I would love to bring you and talk to you some more. Thank you so much.
No problem. All right. So next question, please.
Yes, thank you so much for your presentation, Dr. Yan. I just had one question about mosaicism.
Yeah,
So,
mosaicism. Yeah.
for syndromes that are caused due to mosaicism, does aneuploidy occur in a constant percentage of cells, or do symptoms somehow worsen if more cells are affected by aneuploidy?
Yeah, so that's a very good question. As I told you that if non-destruction occurs in the germline during meiosis, and so after fertilization, that individual will have this, homogeneous distribution of the abnormal chromosome composition in every single cell in that individual. However, sometimes because of the environmental factors, or if you take on some cancer drugs or whatever thing, and you can induce non-destruction in the mitotically dividing cells. And so remember, these cells actually, these are the somatic cells. They are not in the germline. Remember, so that happen only to this individual. So really depending on the percentage of cells within a specific organ that has these abnormal chromosome numbers, the severity will vary. So for example, some individuals, even the Down syndrome, if you see enough number of patients, you're going to see that a range, there's a huge range. And some they show certain symptoms of the Down syndrome, very, very minor. And then those individuals, most likely, you have this somatic, this mosaicism. So like not all the cells contains the abnormal chromosome number. But one thing is for sure, the more cells containing abnormal chromosome numbers, the more severe the phenotype should be. I don't see any other way around. I think at least in Down syndrome case, as far as I know, that's the case. So as I said, some individuals, and they only show some Down syndrome symptoms in certain organs, in certain behavior thing, and then that most likely the causes, the underlying causes, maybe it's the mosaicism. So like the trisomy 21 occurs only in the subpopulation of cells.
Thank you so much.
Thank you. Next one.
I'm not sure if Risha wanted to ask her question.
I do,
Risha.
sorry.
Okay, perfect.
So this kind of goes back to what Ms. Jones was asking you about. So if you were to have an egg that was frozen, and you, like a much older woman, were to have a baby, what are the risks or like damages that can be imposed upon that fetus?
Yeah, so as I said, unless you freeze down your egg and at an early age, and remember the freezing thaw process itself can generate some damage. But these days with technology advancement, and then people can almost guarantee that this whole process and the egg freezing process will bear minimum risk. However, you need in vitro fertilization to fertilize the egg, right? So once the egg is outside in your body, and so how to get the egg fertilized? You have to do it in vitro, meaning that in a petri dish. Okay, so this is a completely in vitro process. And so literally there are two ways of fertilize the egg. Once that you add in a petri dish, you add the sperm into the egg. The petri dish contain the egg, right? So basically add a sperm suspension after, of course, you have to process the sperm. If you get a sperm directly from the ejaculation, they don't fertilize. So you have to activate the sperm, they're going to fertilize. This is called IVF, in vitro fertilization. Or you can use a glass capillary needle. And so like basically inject one sperm into the egg. And then once the fertilization occurs, you culture the embryos for a couple of days before you transfer to the uterus. So remember, freezing thaw process, in vitro manipulation, and then you transfer to the uterus when you are ready, supposedly ready, you are getting older. So that environment and has to be prepared, purposely for the upcoming implantation. So again, so you're going to have the in vitro procedure will introduce problems, and then once they are transferred to uterus, your uterus actually is no longer 21, let's say, like 21, 22, 23. Let's say if you decide to have a baby like at age 45, so that's the 45 years old endometrium. So the environment, microenvironment will change. And so all these things add up, and you're going to have, I'm not saying that, that's every baby born this way will be wrong, but the likelihood you have some abnormalities, either genetically or epigenetically. Genetically meaning that it contains a mutation or abnormal chromosome numbers and structures, and I just mentioned in my lecture. Or epigenetically, just like there's no change in chromosomes, and then so like genetically they're completely competent, but the function of the genes, sort of like got altered, and then that still can bear some risk in the fetus. Does it make sense?
Yes, it does. Thank you, Dr. Yan.
Okay, so next question. We're almost there, but still, let's try to answer your question as much as you can. So one more question, please.
Hi, so I'm sorry if you provided the answer to this question before because my Wi-Fi cut out a little bit. But I'd like to ask that why is it that people with Turner syndrome still have symptoms when inactivation happens in normal females, or I guess what genes are still missing for normal development?
Yeah, that's a wonderful question. Yes, yeah. So remember, in all in all the cells in females, and you have this sex chromosome composition XX, right? And then comparing to male XY, so there's an extra copy of X. And so if you don't do anything, that means the female cells will express twice as much. I mean, like because you have two X chromosomes, and each chromosome carries a certain number of genes, and those genes are going to be expressed in the double dosage, if you will. And then so in order to avoid that, and so there's in the female cells, the X chromosome undergo this so-called X chromosome reactivation. And so one copy of chromosome is not functioning. And then in this way, so you only have one copy of X chromosome that are expressing cells. And then, in the male, the same thing, right? The male, you have XY, so you have genes expressed out of the one chromosome. So her question is very smart. So like in Turner syndrome, you're losing one. And then so that's all like helping you inactivate already one copy, and then those are supposed to be okay. So the matter of fact is that once you are this non-disjunction and you are missing one, you start with one versus you start two. Remember, this X chromosome reactivation is a process. It's the selection and then the timing, and they are all regulated. So in other words, when you have only one copy starting, from fertilization, and they are missing one copy. So in other words, when you have in female cell, you have two copy X chromosome, the one chromosome is going to be inactivated. It doesn't mean that chromosome is not needed. So I definitely think that the facts that the Turner syndrome, they have this problem is because that extra copy, they need to be inactivated in a specific manner at a specific timing. And other than that period of time, this chromosome may be functional. I mean, maybe needed. I think that's what I get. I don't know if Dr. John McCurry is there or not. And so if you can help me with this thing, because Dr. John McCurry knows better about the sex chromosome activation. But anyway, so,
I'm here.
Yeah. So how do you think? Like, Turner syndrome, they're missing one chromosome. And
I
then,
hear you.
Yeah.
So what you said is correct, but I would add to that, some of the genes on the X chromosome escape inactivation. And so normally in female cells, although most of the X-linked genes are expressed from only one copy of the X and not from the other, there is a proportion. It's actually on the order of up to like 25% of the genes escape exon activation. And so females actually express 2 copies. And so in a Turner's individual, they're only expressing 1 copy, which is abnormal. So that's sort of the advanced part of exon activation.
Right. And so in other words, in the normal females, and so yes, they have a extra X chromosome. Doesn't mean that that chromosome can be gone.
It's not
So if
100%
you put that way.
inactivated. It's
Right.
not 100% of the genes that get inactivated.
Right. So the genes escaping the X chromosome reactivation, they're also very important. So when you don't have that thing, somehow you messed up.
Correct.
Yeah.
Okay.
Okay.
Okay,
So-
awesome. Thank you everyone for your questions. They were super direct, pointed. Really appreciate all your curiosity and participation today. And thank you, Dr. Yen.
All
Everyone
right. Yeah.
applause
So-
for Dr. Yen.
All right. So if you have more questions, just email me. All right. Thank you very much.
Thank you so much.
Okay.
Thank you so
Bye.
much.
Thank you.
Thank you.
Thank you.
Bye-bye.